Hello, everybody, and welcome back. Congratulations, you've just run Chip-seq, and you probably have a folder that looks something like this now. And we can see that we started with the design file and all of these, and now Nextflow has made results in a work folder for us, as well as the error and the output that we've been watching. Today what we're going to do is see what data you actually retrieved. And so we could use the terminal to do this and go into results is where you're really going to want to look first. And you're going to see all these different folders. We can go into each one, but you can see this is going to be a lot of changing of directories. So what I recommend is a file transfer program at this point. I'm going to use CyberDuck for Mac. FileZilla works on both any platform. In the GitHub wiki, we have a page on suggested downloads for a file transfer program. So I am just going to switch to that to make navigating this a bit easier. Okay, so now this is that same directory. We can go into results here. BWA is going to be, and we discussed in the previous video what the file types were, and we're going to go over them more and more, but we gave a basic overview of what the pipeline was doing, and now we'll just look at some of the data it provided for us at each of those steps, which is already very useful information. So BWA is where all the goodies are, but first let's check how well the data looked. If you want to look at all of it in sort of an overview, the multi-QC is going to give you all the QC aspects in one HTML file. But if you want to see them individually, you can go into the FASTQ files here and look at each sample report. Let's take a look at one. And so we can see what this guy looks like. Just pull it up in a browser. Okay, so this is already just one sample. What we're going to see is a bunch of quality control assessments of this. And I won't bore you with that, but I suggest looking through all of them to see how your samples look. But you can see right away here that we have read lengths of only 36, but we have 0 poor quality scores and about the normal genome average of GC and a bunch more things. You can click through them with these tabs here. So that's just for one sample, but the multi-QC is kind of nice to look at here in that same report where now it's just downloading to my computer with the file transfer. We can take a look at it. And so what's nice here is we're going to see all 3 of our samples. Our input is always going to look a little different, and that's good than our actual controls. And you can scroll over these and it gives you this information, and there's just tons of data already that you can present at lab meeting or wherever that on the quality of your Chip-seq. So that's handy that all of those things would be done in different programs and different output files for each sample, and Nextflow's put this all together for you so that whatever you've run, it'll put it all together. And you can watch a video tutorial on other aspects of this and go through these different files and get familiar with the file types and the sort of metrics that we briefly went over in the previous video that that Nextflow is making sure to use programs to make sure your data is high quality. Okay, so that's just the quality control stuff. That's usually the first place you're going to want to look. What's nice is the pipeline info has all been tracked for you, what time it was run, how it ran, what versions were run. You have a whole file that tells you what versions were run. So this is where the reproducibility comes in with Nextflow. It took care of all this for you. TrimGalore is going to give you its output specifically. It's one program that was run. I'm going to skip that so we can get to the good stuff. So when you go into BWA, you're going to get merged library. And we talked about SAM tools and these things that were happening right after the alignment to sort of figure out how to put the reads from alignment into these other programs. But Max is the program. So there's 2 folders that are really useful here. They're all useful, but the 2 most useful are the big wig. And what that is it's a representation of the BAM file that's smaller So you can see the raw sequencing reads. And that's what we're going to do next is take some of these and go look at them in the IGV browser in just a second. But bigwigs are great because then you can see the raw data. And then Max is the peak calling program that said, I saw more signal here than the input, and tells you all of the regions of the genomes, or ranges, as we're going to start to talk about in those terms, all the ranges in the genome where there was significant signal. So let's look at Max first, and we'll look at broad peak. So you'll see you have now a peak file for each of your replicates of ASH2. Obviously, the input is not going to call peaks, so you're only going to get peaks for these because input was taken into consideration. But in the bigwig folder, you will see that the input's here, and that's really nice to visualize. What does your input look like? Because sometimes you have to worry that peaks can be caused where there's low representation in the input. So the denominator is getting lower, so the likelihood of significance gets higher. So, so what you'd want to do now to go visualize these is we're going to look at the bigwigs and the Max. And so what you would do is go in to Max and take, what we're going to look at is broad peaks are easily readable by IGV. There's gap peaks and annotate peaks. You can read more about those. But really, if you want to do this in Excel, you can. I don't recommend that. But what we're going to do for visualization purposes is take these files from your file transfer program or from your local computer and drag them to your computer. And I've already pre-cooked a nice folder for us to explore where I've just gone through and I went and got these same, we should actually have identical data, and we'll probably see that as well. So if we look here, I already have a folder with the, don't worry about this one. There's the input bigwig, the ASH2 consensus peaks, broad peak, and these are all in here. So you're basically going to get the broad peaks. Don't worry about the consensus peaks for now, but essentially what is in there is a file that has the two replicates merged and said, this is where all of the data overlapped. And we'll get into that. But for now, let's just look at each sample individually and see what peaks were called. So gather up these two, and then in the bigwig folder, gather up these three bigwigs, and we'll take a look at them. And just for fun, what I did is ran these same samples that we just ran together on two different versions of the NF Core Chip-seq pipeline. So we can compare to see what would happen, how would the bigwigs and the peak calls look differently if you ran it a year and a half later, which is what we just did since the last class. And I'll leave you in suspense of that. So for now, what I'll do is introduce you to IGV. So once you've made a folder with your bigwigs and peak files that you're interested in putting into IGV, we'll go, the link is in the description, but you can just Google IGV browser download, and what you'll find right away is here. The links are in the syllabus again, if you're using Mac, Windows. I tend to use the ones that were Java's already included, so you don't have to change your Java settings here. So you would just download one of these, and then now you'll have an icon that looks like this, like IGV shown here. And what you'll see at first, it's going to end up looking like, so what you're going to see here is just sort of a blank pane once you've installed IGV. It already has the genes, and it tells you which genome you're looking at here. We're looking at HG38, and that's where our data is. But if you are working in mouse or other things, there's a lot of data in there. But for now with HG38 is where we're going to start, and I'll show you a little bit more of that. Here's where the coordinates are going to be, but for now, let's just load our files. So file, load from file, and I'm going to go to my handy, already pre-cooked class folder. I'm just going to select all of these that I want and hit open, and boom, there they are. And so here in this box, you can type in the name of your favorite gene. Let's say it's fire, which is mine. It'll take you right there. And if you want to move left and right, you just simply hold down with the mouse and you can scroll once that hand shows up, and you can start to see peaks. So here's those tracks where the peaks were called. I'll show you this in a more organized way in a second, but it's just nice to know that you can already see right away from your data that your input here doesn't look like this peak is showing up, and Max accurately called these two different peaks in the two different replicates. So you can quickly browse around there. You could actually just add in the coordinates you're interested in or your favorite gene. And then you can also export a given image by you can run here, you can hit save image, and it'll give you options for how to save this browser, and where you might want it as an SVG so that you can use it in a vector. Otherwise, you can save it as a ping, and so on. We can load some information here. So if you go to file load from server, IGV has some data already uploaded on it that you can access. Unfortunately, the older genomes have more information, but you can look here in annotations, and let's say we want a different version of genes, or let's say we wanted the repeat regions. We're interested in human-specific transposons. Let's pick LTR, so we want to see that. You click there, and now that track is going to show up on the bottom here, where all the LTRs are shown here. Okay, so if you go to older genomes and want to work in them, like let's say HG19, and you're going to lose your current session, but you can now go look at this file, load from ENCODE, and look at all of this data. So you might want to load your tracks or convert them to HG19, and you just have massive amounts of data. A lot of this is also in the UCSC genome browser, so we'll go over that in a different video, and there's different considerations. IGV is just a great way when you get your data to quickly visualize it, see if your replicates have similar peaks, etc. And so those are some of the main features: file load your files, and then load from server. And then browsing around is pretty straightforward, when you can just go to your favorite gene, GAPDH, and if you wanted to scroll left and right, you just hold down, and the faster you hold down, the further it'll scroll for you. So those are really the main features of IGV, just a quick quality check. And how simple and easy it is to go right from your Nextflow output and loading it into a visual browser. I apologize, I can't make this much bigger, but I'll try and show you that this, the first line is running it on the old version of Chip-seq. This is a bigwig file for this interval here with fire, a gene we're interested in. And you can see both replicates called the exact same peak, essentially. So that's really good news, that if you run the old pipeline and the new pipeline, you get the exact same bigwig, which you'd hope the alignments would be the same, but you also get the same peaks. So the algorithm hasn't changed that much. Same for the second replicate, run on the old version, new version, and the peaks, and you can see the structure of the peak is not present in the input. I always like looking at the input to see what's going on. You'll also notice that IGV auto scales these tracks for you, so you can right click in the corner and you can change this to a heat map if you want to see the signal that way, shown here. So now you can sort of see that signal a little more strongly. And change this to a heat map, and you can see that that blue is really strong here compared to there. And so you can play around, and also the track height is important for scaling samples. It'll automatically scale for you, but you might want to look at the data all at the same scale, and you can change it in there. So I think that's it for this video. That we've now run our first Chip-seq, and we just took a peek at it, ran it in an old version and a new version. We got the same thing. So this nice test example worked pretty good. It'll be interesting to see if you got the exact same data. I'm guessing it'll look identical. And then next we will start moving forward, looking more deeply at the data in the following classes. So have fun exploring your data, looking at your favorite genes, pulling up annotations, and in the meantime, code light.